All right, so today I'm going to be presenting just one aspect of this multi-center project, looking at the survival and virulence of Listeria monocytogenes during long-term storage on low moisture foods. So just a quick disclosure, the study was supported by ILSI North America, which supports myself as a student at the University of Guelph, and the authors have no financial relationships with any commercial interests related to the content of this study. It's just a very brief table of contents. I'm going to be discussing some background on the project as well as our main objectives and how we achieve these objectives in addition to results, and closing with a summary of those results and the main takeaways of this work. So I hardly have to impress upon this audience how important Listeria monocytogenes is as a foodborne pathogen. It's capable of growth over a wide range of temperatures as well as pH values and is highly salt tolerant, capable of survival in up to 20% salt. Now, while listeriosis is quite rare, it is associated with a high case fatality rate of about 20%. And this was especially demonstrated in the most recent large-scale South African outbreak that we saw of listeriosis between 2017 and 2018. So low moisture foods are those products that are defined as having a water activity below 0.85. And a lot of these products are ready-to-eat foods. So in Canada, in addition to the EU, we do have an action level of 100 CFU per gram for Listeria monocytogenes in these products which do not support the growth of the pathogen, as well as its required absence in those foods that are targeted towards high-risk populations. Here in the US, there's a zero tolerance for Listeria monocytogenes in these products. Historically, LMFs, I'll be referring to low moisture foods as LMFs, have not been considered a food safety concern. However, in the past decades, we have seen numerous outbreaks related to these products, particularly concerning Salmonella, Clostridium, E. coli 0157, as well as Chronobacter. So in the past 6 years or so, there have been several recalls of low moisture food products caused by the contamination or the potential contamination of these products with Listeria monocytogenes. And Table 1 just gives some examples of the products that were recalled in Canada between that time frame of 2013 and 2018. And on the far right, you can see the number of individual notifications for each of these different regions of the world during the same time period for which there were recalls of these products. And it's important to also be aware that the majority of these recalls are associated with nuts, seeds, or their derivative products. So there have been a number of studies that have established that Listeria monocytogenes is very capable of long-term survival on low moisture foods. And just two examples that I have up here are its long-term survival in pecan halves as well as wheat flour. And we see in these two products that we do have survival for up to about a year. So the contamination of these products with Listeria monocytogenes is a concern as a potential vehicle for the introduction of these products, or for the introduction of the pathogen, into foods that may support the growth of the pathogen. It's also important to gain a better understanding of how Listeria acts in these foods because the number of recent listeriosis outbreaks have been associated with unusual vehicles and potentially low dose response. So the two core objectives of this study were to examine the survival of Listeria monocytogenes during storage on a wide variety of low moisture foods, which were discussed previously, at two storage temperatures, so refrigeration at 4 degrees Celsius, and then room temperature, which we've defined for our studies as 23 degrees Celsius. We're also interested in assessing the virulence of the pathogen both before and after long-term storage on these foods. So this was done by preparing a 4-strain cocktail of Listeria monocytogenes. And then, depending on the food, they were either dry or wet inoculated. So for the dried apples, raisins, and dried strawberries, the foods were dry inoculated using a sand carrier that was contaminated with the pathogen. For the chocolate liquor, the cornflakes, as well as the pistachios, these products were wet inoculated, with the former two being mist inoculated in order to maintain the structural integrity of the products. And there's just a picture of the apparatus that we used up above there. So the wet inoculation was followed by a drying period, as well as an equilibrium period to allow for the water activity to come back or to stabilize, before the foods were stored at those two temperatures that I previously discussed, along with their respective relative humidities. So during the storage period of a year, the populations of Listeria monocytogenes was quantified using both culture-dependent and culture-independent methods. So the use of selective versus non-selective agar was really dependent on whether or not there was a pre-existing microbial population on the foods, which was the case for the dried fruits as well as the pistachios. And then we also used a viability qPCR method to selectively quantify the live cells. And so this live cell population may be including both the culturable cells, but also cells that are not culturable but may still be alive. We were also interested in looking at the relative survival of 4 serotypes of Listeria monocytogenes. So the 4-strain cocktail was composed of 4 different serotypes. And we did this by using a multiplex PCR method, which I'll be describing further in detail further in the presentation. And lastly, the virulence of the pathogen at both at the beginning and end of storage was assessed using an in vitro CACO2 cell model. And we looked at the attachment, the adhesion, as well as the intracellular replication efficiency of the pathogen. So the results can pretty much be separated into 2 broad groups based on the objectives of our study, looking at the survival and then the virulence. So in the dried apples, which were dry inoculated, we were actually unable to recover and enumerate Listeria. And initially we thought that this may be due to the presence of sulfites. However, a discussion with the supplier turns out there's no sulfites. And the pH we determined to be about 3.7. So something is happening here that is different than on the raisins, for example. We see a very sharp decline in populations at 23 degrees Celsius, which is in the black. And then we see a slow decline in populations at 4 degrees, which is in blue, with populations remaining above the limit of detection by the end of storage, which is indicated with the dotted line. On the dried strawberries, we see a very similar story. So we have a rapid inactivation at 23 degrees Celsius with a slow decline at 4 degrees Celsius, about double the rate that we saw on the raisins. And again, populations were above the limit of detection by the end of storage. On the chocolate liquor, we saw enhanced survival at 23 degrees Celsius compared to the dry fruits. So we did see rates of decline at about 0.8 log CFU per gram per month, with populations falling below the limit of detection after month 8. And then again, a very slow decline in populations at 4 degrees Celsius. Very similar survival dynamics were observed on the cornflakes. We see very slow decline at 4 degrees Celsius. Again, we see populations on the cornflakes stored at 23 degrees Celsius falling below the limit of detection after month 8, very similar to the chocolate liquor. And there are actually 2 rates of change given there for the population on the product stored at 23 degrees Celsius because a biphasic model was fit to this curve. So within the first 4 months, we see a very rapid decline, about 0.8 log CFU per gram per month. And then after 4 months of storage, the rate of decline decreases to about 0.2 log CFU per gram per month. Lastly, on the dry roasted pistachios, this was the only food product for which we saw both, in both storage conditions, populations of Listeria monocytogenes remaining above the limit of detection by the end of storage. At 4 degrees, we actually see no significant decline in populations, and the populations on the product stored at 23 degrees Celsius were only decreasing at about 0.3 log CFU per gram per month. So all of these long-term survival assays were done using culture-dependent methods. And so we were curious in seeing if the decline in populations that we saw were due to cell death or whether it was more of a transition into a cell state where the cells are no longer culturable, but they may still be viable. So we looked at the viable cells versus the amount of culturable cells after storage of Listeria monocytogenes in these foods at 4 degrees Celsius on the chocolate liquor, on the cornflakes, as well as the pistachios. So you can see populations determined using TSA in gray, as well as PMA qPCR in red. PMA, or Propidium monoazide, is commonly used to selectively quantify live cells, at least considered alive by membrane integrity. And we can see that populations quantified by PMA qPCR are greater than that obtained by TSA by the end of storage for all 3 foods. And after long-term storage, populations quantified by PMA qPCR were 1.8 to 3.7 log CFU per gram higher than that obtained using culture-dependent methods. Moving from a cocktail level to the strain level, we examined the effect of storage on the relative survival of 4 serotypes that made up our cocktail. And so the serotypes included in our cocktail were half A, half B, 3A, as well as 4B. And this was done by multiplex PCR on individual colonies recovered from the foods both at time point 0 as well as the end of storage. For populations recovered from samples at 23 degrees Celsius, the rates of decline were much faster than that at 4 degrees. So we actually ended up, instead of taking colonies at the end of 1 year, we chose time points during the storage period at which there were a lot of colonies that we would be able to pick from the plates, rather than at the very end where there may only be 5 or 6 colonies. So we saw on the dried fruits a significant decline in the relative survival of serotype half A, as well as a significant increase in the relative survival of serotype 3A. And this was seen on both dried fruits. And it's also very interesting to note that the levels of serotype half B and 4B were quite low. Looking at the chocolate liquor, we see again a decrease in the relative survival of serotype half A with both storage at 4 degrees Celsius as well as 23 degrees Celsius. Again, we see an increase in serotype 3A with storage at 4 degrees Celsius. And for the first time, we saw the increase in relative survival of serotype half B with storage at 23 degrees Celsius. On the cornflakes, we saw the opposite. So we saw an increase in the relative survival of half A with storage in both temperatures, and no effect of storage for all of the other serotypes that we tested. On the dry roasted pistachios, we again see a decrease of serotype half A at both storage conditions. Again, an increase of serotype half B at both storage conditions. And for the first time, an increase in the relative survival of serotype 4B after storage at 23 degrees Celsius. So we've established that Listeria monocytogenes is perfectly capable of surviving for up to a year in these products, but it's still virulent after the storage period. So we assessed the virulence of the bacteria we were able to get back from these foods using a CACO2 cell model. So cells were grown or CACO2 cells were grown until they were semi-confluent, and then starved for 24 hours before infection. Here we infected the cells with Listeria monocytogenes harvested from the food washes and then resuspended in cell culture media. And this was done at a multiplicity of infection of 10 to 1, Listeria monocytogenes to CACO2 cells. 30 minutes post-infection, we harvested the Listeria monocytogenes cells, and we recovered them on TSA-YE to determine the attachment efficiency. After 2 hours post-infection, the monolayers were washed with gentamicin in order to kill all extracellular Listeria monocytogenes, and then the CACO2 cells were lysed with Triton X, and then the pathogen was recovered again on TSA-YE to determine the invasion efficiency. 6 hours after that, so this brings us into 8 hours post-infection. The CACO2 cells were lysed, and then Listeria was recovered on TSA-YE again. So we can see the virulence of Listeria monocytogenes on after storage on dry roasted pistachios at 4 degrees Celsius. So here we have the attachment efficiency of samples at time point 0, which is indicated here with month 0, so the third column, and then at the end of storage, which is the fourth column, as well as 2 controls. So LM is the cocktail resuspended in cell culture media, and then LM plus P is the cocktail being inoculated into the food matrix and then being recovered immediately for virulence assessment. So we can see that attachment efficiency increased in the presence of the food matrix, but looking at the invasion efficiency and the total virulence potential, which was determined from the intracellular replication percentages, what we saw was little overall effect on Listeria monocytogenes virulence, at least compared to the cocktail control. So the virulence of Listeria after storage on the chocolate liquor at 4 degrees Celsius, we see that, again, attachment efficiency increases in the presence of chocolate liquor. However, these effects appear to reverse after long-term storage. The evasion efficiency appears to decrease after the drying equilibrium period. So again, we see an increase, after the drying equilibrium period. So just summarizing all of those results, we have Listeria not surviving on the dried apples. At 23 degrees Celsius, we see populations declining significantly by greater than 5 log on cornflakes and chocolate liquor, and greater than 4 log on dried fruits, and 2.6 log CFU per gram on dry roasted pistachios. The linear rates of decline for both for populations stored at both 4 degrees and 23 degrees Celsius are given here from greatest to least, and we can see that rates of decline were the greatest on the dried fruits and the least on the dry roasted pistachios. Quantification differences observed between plate counts as well as the PMA qPCR suggests that there's an existence of a viable but non-cultural population of Listeria on these foods. Serotype 4B, at least the strain that we used in our cocktail, may be particularly sensitive to desiccation stress, while serotype 3A appeared to be the most resilient. And it's also important to note that serotype 4B increased only during storage on dry roasted pistachios at 23 degrees Celsius. And the relative concentrations of the relative survival of serotypes on the cornflakes, with the exception of half A, these stayed the same throughout storage regardless of temperature. Looking at the virulence, we saw that attachment efficiency increased in the presence of the food matrix for both after both storage on the dry roasted pistachios as well as the chocolate liquor. However, on the chocolate liquor, the effects reversed after long-term storage. And the invasion efficiency also decreased after the drying equilibrium period, so that would be time point 0. So concluding, Listeria monocytogenes is very capable of long-term survival on these products that we looked at, especially with storage at 4 degrees Celsius. And serotype 4B may not be a serotype of interest for these products versus a serotype that's sort of considered lower risk, or 3A. And there is evidence for a viable but non-culturable population during long-term storage on the chocolate liquor, the cornflakes, as well as the dry pistachios. The attachment efficiency of Listeria monocytogenes may be enhanced in the presence of the food matrices that we tested. However, long-term storage on the products did have little overall effect on virulence, at least compared to that of the original cocktail. The chocolate liquor, however, we did see lowering the invasion efficiency of the pathogen. I'd just like to acknowledge our team at the University of Guelph, so my advisor, Dr. Jeff Farber, our research manager, Dr. Valeria Pera, as well as another student that we had working on this project, Ryan Kuzi. I'd also like to acknowledge our collaborators at Health Canada as well as NC State, and of course, ILSI North America for financial support, which allows me to come to conferences like these. So thank you very much. So my contact information is up here if you have further questions and we can't get to your questions today. And I'll open the floor to questions.
Hi, Vivian. Thank
Hi.
you. Jennifer Aka from Virginia Tech. Can you share a little bit about some of your strains, if they were clinical isolates or if any of them were outbreak isolates from low moisture foods?
Yes, so because there have not yet been any outbreaks of listeriosis related to low moisture foods, we don't have any of those isolates to work with. There also have not yet, at least to my knowledge, any confirmed cases of listeriosis due to the contaminated LMFs. However, the strains that we did use, so half of them were food isolates and the other half being clinical isolates. The closest we could get, at least looking at the foods that we actually looked at, was we used a chocolate milk strain that was from an epidemic in 1994. The other strains that we did use were a food isolate from the Mexican-style cheese outbreak of 1985. We used a human sporadic isolate, which was our serotype 3A strain, and then another clinical strain from the hot dog outbreak in 1989. Yeah. More questions? Okay, thank you very much.